Hi all, and welcome to my virtual poster presentation for the CURAS Research and Scholarship Fair. My name is Sydney Dang, and I'm a senior studying biology and studio art. My research project with Dr. Peter Stager's lab focuses on the cellular pathways in aminoglycoside-induced hearing loss. So let me get to the next slide. There we go. Aminoglycosides are some of the most successful drugs in treating gram-negative infections, such as meningitis, endocarditis, or respiratory infections. This class of drugs is also used as preventative measures in preterm infants to prevent sepsis and to treat patients with cystic fibrosis. Many reasons for the use of aminoglycosides includes chemical stability at ambient temperatures, lower incidence of microbial resistance, and relatively lower costs compared to synthetic versions of this medication. Aminoglycosides do have their drawbacks, as systemic administration of these drugs can cause acute kidney damage and/or permanent ototoxicity. So of patients receiving intravenously administered aminoglycosides, as many as 20% of these patients will experience aminoglycoside-induced ototoxicity. Aminoglycosides are damaging to the inner ear and cause permanent hearing loss because they target the outer and inner hair cells of the cochlea that detect sound. Once hair cells become physically damaged and are no longer functional, they are often phagocytosed by adjacent supporting cells. New hair cells do not regenerate in place of the phagocytosed ones, and thus hearing loss is permanent. Most studies regarding aminoglycoside-induced hearing loss come from studies of healthy subjects that do not have systemic inflammation. This is a key distinction, as aminoglycosides are typically only administered to patients with severe infections and hence inflammation. Inflammation causes an alteration in the blood-labyrinth barrier, which increases the transport of aminoglycosides into the inner ear. This enhances the degree of hearing loss experienced by those individuals. So in this project, we'll be using MDC T-cell cultures without the use of antibiotics and introduction of an inflammatory agent, lipopolysaccharide, to mimic the physiological responses to systemic inflammation. So the proteins of interest for this project are MyD88 and TRPV1. I want to verify that cytosolic MyD88 sensitizes TRPV1 activity. TRPV1 is an aminoglycoside permeate channel that is activated by factors associated with inflammation and pain and is expressed in key aminoglycoside trafficking locations of the inner ear. So to examine co-localization of these two proteins of interest, we used PLA, or proximity ligation assay, and found that MyD88 and TRPV1 do co-localize before LPS treatment, as depicted by the red puncta. Here you can see the red, and that's found within the cell's cytoplasm, and the blue staining indicates the cell's nucleus. To see how MyD88 and TRPV1 interact with each other, we plated MDTC cells and gave them either no LPS or LPS treatment for 5, 30, and 60 minutes to activate TRPV1. Western blotting was then performed to see how TRPV1 activation with LPS impacted protein expression. At 10 micrograms of total protein loading, there was a significant increase in MyD88 expression at 5 and 60 minutes, which are consistent with previous findings. From our PLA and Western blot, these data suggest that LPS exposure upregulates MyD88 expression that may potentially influence TRPV1 activity and subsequent cytotoxicity. So future studies would include continuing our PLA co-localization studies of TRPV1 and MyD88, but under LPS conditions. We want to study how toll-like receptor 4, or TLR4, a receptor that triggers inflammatory signaling cascades in response to infection, interacts with TRPV1. So due to COVID-19, it is difficult to be physically in the lab during the summer months of 2020, and some of the goals outlined in my original proposal were not fulfilled. However, I am very ambitious and excited to continue this research after graduation. And finally, I would like to thank Amy Schreiner for teaching and assisting me through the laboratory protocols of cell culture, PLA, and immunoblotting. Also, a big thanks to Amy and Dr. Stager for reading through my materials and offering revisions along the way. And a final thank you to the CURAS Surf program for sponsoring this work. If you have any questions, please find me at my Zoom or email me at sydneydang@creighton.edu. Thank you so much for listening and have a great day.